Hello, my name is Chelsea Walsh, and I'm going to be talking to you about the where and when of earthworms in wheat fields. I'm also going to add in some what and who just to keep it balanced. And the reason that I am looking at earthworms in this region is because there's a lot of research that suggests that earthworms can have a really positive impact on crop production. They increase nitrogen cycling, they can increase water infiltration in some cases, and improve soil aeration. But our question is, in our particular climate, which is going to be limited because we have that xeric soil moisture regime, which means that we don't get a lot of rain in our summer growing period. We think that this may be limiting the impacts, and we're trying to figure out exactly what those limits are and what role earthworms may have to play in our current climates across the climate spectrum we see in our region, as well as potential future climates that we might see in this region as moisture and temperature patterns change. So I'm going to start with the who of earthworms. Who is here? And by who, I really mean what species do we have present in this region. There's some previous work in this region, and that has found 4 species: Aparectidia trapezoides, Aparectidia tuberculata, Aparectidia turgida, and Lumbricus terrestris. And of these, Aparectidia trapezoides is by far the most common, especially in agricultural fields. And you can see on this map here, they covered a lot of region, a bit more than I'm going to cover in my research. But in those agricultural fields, trapezoides was dominant. And so we decided to follow this up with a more concentrated look at just agricultural fields in just this dry land, wheat-producing region of the inland Pacific Northwest. And so we looked at a total of 36 fields in 3 different cropping zones. There was the crop fallow zone, which here you can see is the yellow square boxes. We also looked at a transition zone, which is moving towards the annual cropping zone, where there's enough soil moisture to be able to grow a crop every year. And so we looked at those 36 sites, and in the survey in the 3 years, we found a total of 2 species, so 2 less than found before. And of those, by far, Aparectidia trapezoides was the most common. I found 1 adult individual of Lumbricus terrestris. However, it is important to note that our sampling methods are going to underrepresent Lumbricus terrestris. This is a species that burrows up and down. You're probably familiar with them, night crawlers, the really big worms. And those ones, with the way I'm sampling, which is just basically digging holes and then looking for worms in the dirt, you're just not going to find them because they really do hear the shovel and retreat down in the soil profile. And so it's more uncommon to be able to sample them with the method I'm using. However, you can usually tell if they're present. You can see their holes and their castings on the surface, and there have been a couple fields where I saw some pretty strong evidence that they were there, but really the majority of them really didn't have any indications that that species was going to be prevalent. This is a picture here of Aparectidia trapezoides. Unlike Lumbricus terrestris, this species makes horizontal burrows. It stays within the mineral soil and actually consumes the mineral soil itself. Other earthworm species eat the litter from above off the surface, but this one actually stays in there and just eats the soil itself, breaking down the organic matter, which contributes to its ability to really improve crop production because it's consuming that organic matter, it's releasing nitrogen in a much more available form. Its casts have a lower carbon to nitrogen ratio than the bulk soil, and so this is thought to be the main reason they're affecting crops is by improving fertility in the soil. You can see it's about 10 centimeters long. This one is maybe on the bigger end of them. They do get a little bit bigger than this, but they really aren't near as large as the night crawlers. And so we know that who is here? It's mostly trapezoides. And so then the question also was, across this zone that has this steep climatic gradient, where we see a big range in cropping practices and soil moisture, where are they in those fields? And so we found them in 21 of our 36 fields. And as you can see, they're fairly geographically segregated. You have our drier zone here, where we're moving into our crop fallow zone, and then we have our annual cropping over there towards the east. And so we really find them mostly in our annual and transition zones. The one interesting example is this dot down here near sort of northeastern Oregon. That represents where Pendleton Station is, and at that site we had a long-term conventional and no-till sites paired right next to each other. And when we sampled in our no-till sites, we found earthworms, and when we sampled in the conventional, we didn't. Most of our other sites in that region are no-till, and so it could be that if we found more conventional till sites in that region, we would also not find earthworms in those sites as well, probably because the higher disturbance and generally the no-till sites retain that soil moisture a little bit better, so they're going to stay a bit wetter. So the next question was what limits their distribution? And when we looked at these sites and the 21 that had earthworms and the rest that didn't, we found a nice even threshold right around 350 to 400 millimeters of mean annual precipitation, where the sites that we sampled below that did not have earthworms, and the sites that we sampled above that, all except for that one in Pendleton, all had earthworms. And so it seems like at this large scale, it's going to be precipitation and just the availability of moisture which allows them to succeed. So now that we knew who was here, where they were, and what was determining this broad scale distribution, what we wanted to narrow in was on their seasonal activity. So in this region where we have a xeric soil moisture regime, we wanted to see what period of the year did they have adequate soil moisture and good soil temperatures to be able to be active. And then this figure, I've sort of made my best guess on what periods of the year may have limited earthworm activity, and that's those parts highlighted in gray. And as you can see, that's really a very large portion of the year. Earthworms are generally thought to become inactive below 5 degrees Celsius. They don't necessarily die, but they also aren't moving around, they're not doing anything, and obviously it'd be very hard for them to burrow through frozen soil. And so this picture on the right here is a picture of an earthworm curled up into a little ball inside a spherical chamber, and that's known as estivation. And this is one of the responses this species, Aparectodia trapezoides, can have to really hot, dry conditions. It curls up in this little ball, it makes itself a nice mucus-lined spherical chamber, and then it goes in there and it curls up, slows down its metabolic activity, and kind of waits around for the soil to get nice again. And so this is a picture of an earthworm doing that. Really impressive. They can come out of this estivation extremely quickly. When we sample, we take them and we put them in a plastic bag with a wet paper towel, and they really get moving around pretty quickly after that. So to look at this question of activity period, we picked a smaller set of 6 sites to sample more frequently, about every 2 weeks. It would not be practical for us to go out to 36 or even 21 sites every other week. So we picked these 6 sites, and there are 3 of them clustered in a precipitation zone of about 450 to 500 millimeters of mean annual precipitation. And those are kind of very close to each other, sort of within a 2-mile radius near Colfax. And then we moved over to the east, and we have a site we're looking at Cook Farm, which is near Pullman. And we have a site that is practically in town in Moscow. And then we have another site down by Kendrick, and these are all higher rainfall zones. And so we're hoping that maybe we'll see some differences between these 2 areas in terms of when earthworms estivate and what temperature-moisture combination that they're responding to. So this is one of my data loggers that I put out in each of the 6 fields. I put it out to measure soil moisture and soil temperature on a continuous basis, so I could catch high temperatures, low temperatures, and any unusual soil wetting events. And I had those sensors down at 20 centimeters. I also had a rain gauge in there just to, my previously mentioned precipitation ranges are based on model data, not on any actual weather station. And so this was to maybe capture actual precipitation differences between your sites. And so here on April 10th, you can see the day that we installed these. That was the first day I sampled. The soil temperature is still fairly cool, staying below 12 degrees Celsius throughout the day. And soil moisture was still pretty high at 20.2%. But as you can see, we didn't capture quite as early in the season as we would have liked. The soil starts out much wetter than that. And so over the next year, we're hoping to catch it a bit sooner and get the beginning of earthworm activity in the spring. On the next slide, I have a picture here of the same site on July 30th, and that's when I took the data logger out, because that was right before farmers harvested, and I didn't want this to be in the way of their combines because it's not good for their combines and it's not good for my equipment. And so that's the point when we had the soil moisture sensors and the soil temperature sensors out. And so we haven't had them in the ground since then. And I am waiting to put them back in the ground until I hear from the farmers that they are done with their tilling operations, their planting operations, whatever they need to do this fall, so I don't have to worry about them being in the way throughout the winter. So here you can see that the wheat is mature, it's really dried down, it's ready for harvest, and the soil moisture is down all the way to 6.3% moisture, and the soil temperature is quite high with a high of 28 degrees Celsius and a low of 19 degrees Celsius. In the earthworm here, you still see a fairly high, at this site anyway, biomass, but all that is estivating earthworms. And what this really means is that at this site we probably happen to find an adult earthworm estivating. The adults are much heavier than the juveniles, and they really make the biomass a lot higher. So this might be a fairly high biomass, but could still be a fairly low density. And so then here we have a picture of when I went out on October 30th, once again to the same site. And you can see here it's been harvested and the wheat's gone, and we've got some residue. This happens to be a long-term no-till site, and so this particular site will not be tilled. I do have some sites that have a full range of tillage, from reduced to minimum to no-till to a fairly high tillage rate. And so here we found that the soil moisture was still quite low. There's a wetting front down to maybe 5 to 15 centimeters depending on your site, and above that wetting front our soil moistures were probably around the 12 to 13% volumetric soil moisture range, but then once you got down below it, they're still really quite low, and that's where you're going to find those earthworms. Earlier in the season they moved down so that they could get down to where there's more moisture, and then they started estivating when that dried out, and now the water still hasn't reached them, so they haven't really realized yet that conditions are getting better up higher, and so they're still curled up and estivating. And so here are some of my results so far for biomass, and these are averaged for the high rainfall sites together and the low rainfall sites together. And you can see that this red line I have in here, that's the point in sampling when they started estivating. That's some point in that time between those two sampling sites. Basically, activity pretty much stopped. It went from a point where all of the earthworms that I collected were active, and then it went to a point when none of them were. And so somewhere in that 2-week period, they all just got too dry and they quit. So before that, you can see that another interesting thing is that biomass, and if we look at this next slide, also density was higher at our lower rainfall sites, which was a bit of a surprise. These were lower rainfall, their soil moisture was lower throughout the season, and yet they actually had more earthworms. My current working hypothesis is that that warmer temperature is giving them a higher activity rate and a higher reproductive rate, a faster developmental rate, and so we're ending up with more earthworms, even though maybe there's not as much moisture, but there's still enough moisture. And then you see that right about the beginning of June, they all start to estivate and we don't have activity anymore. And so at least in this first season, we found an active period of about April and May. I'm sure that they were active before April because our densities were already quite high, but that's really not long into the summer where we still have earthworms being active. And so here, this is just my soil moisture and soil temperature data to give you an idea of what's going on with the soil conditions to kind of compare back to those earthworm densities. And so that red line is still that time when we found the earthworms to be estivating. And you can see that by that point, the soil moistures were dropping down, but you can see that that teal color, that's the soil moisture in our higher rainfall zone, the big block of teal, and then we have the block of green, and that's the soil moisture in our low rainfall zone. And you can see they had started to drop a good bit, and we're down to about 10% and actually still as high as 15% in the high rainfall areas. And yet the earthworms are starting to estivate. And our thought is that that's probably related to somewhat to the soil temperature, because at that point soil temperatures at the high points were getting up to 18, 22 degrees Celsius, which is really a more stressful temperature for them. And so we think it's going to be important to look at this combination of our soil moisture and our soil temperature to really find out what is triggering them and to go into estivation, so that hopefully you can take these sort of numbers and apply it to a larger area, to a broader range of conditions, and find out what sort of active periods do we have for earthworms in this region. And so our next step will be to continue to collect this data to get more data points for soil moisture and temperature conditions and what the earthworms are doing. And we can see here some strategies we're looking at using to analyze this data, and one of them is looking at these soil moisture and temperature combinations and what earthworms are doing. And so my open boxes are boxes, those indicate when earthworms are estivating, and the filled in ones indicate when earthworms are active, with the green still being the lower rainfall site and the teal still being the higher rainfall site. And so you can see there seems to be, it seems to be more oriented towards soil temperature, but there does seem to be a soil temperature moisture combination sort of boundary where we stop seeing earthworm activity at around actually only about 12 degrees of Celsius, and then about somewhere between 10 and 20% soil moisture we see estivation. Another thing we're considering is because these single point data for moisture and temperature don't reflect any of the previous conditions, which are very important for the earthworm. They're not responding on a instantaneous basis. They're responding to what's happened to them in the time previous. And so one thing we are thinking about looking at is some sort of cumulative soil temperature and possibly cumulative soil moisture. And so this one here, I'm tentatively calling cumulative soil heat hours. And this is looking at the number of hours that the soil temperature has been above 15 degrees Celsius, which is generally, from lab data, considered to be a temperature that's stressful for earthworms, where they start to show weight losses and reduced survival. And so what you can see here is that it's really not a linear response at all, and it's really not very many cumulative soil heat hours before we start seeing estivation. It seems to happen pretty quick. Another one we're looking at is just this relationship of moisture, which we expect to be a big predictor, just because earthworms require soil moisture to breathe, to maintain their own soil moisture balance. Earthworms don't have a lot of great ways to prevent water loss to their environment, and so having adequate soil moisture is very important to them to not dehydrate. They also breathe through their skins, much like a gill works in a fish. And so they require that soil moisture to be able to exchange carbon dioxide and oxygen with the environment. And so without that soil moisture, they can't breathe. This is the same reason earthworms come to the surface when it gets too wet. The water gets deoxygenated and they start to drown. So they come to the surface. And here you can see on this graph, it's somewhere around, here it looks to be about 15%, but obviously we have some overlap here, and so I think that's where we're seeing that soil temperature and soil moisture interaction. So in summary, Aparectidia trapezoides was the primary species we identified in all of our work so far in this region. Lumbricus terrestris may be underrepresented, and in some fields there's evidence from their burrows that their populations might actually be quite large, but from previous work and from my own experience, it seems like this might not be a large part of the region. It seems to be a lot limited to slightly higher rainfall areas and reduced disturbance sites. And so they may be very important in some sites, but across the region, their influence may be limited. Their estivation in this year began in early June and still seem to be inactive as of October 30th. And so we're looking forward, I guess, to seeing when they start to get moving again and have a start point for the activity and see how long they last into this upcoming cool temperatures. We're expecting that interaction between soil moisture and soil temperature to be the big determinant in when they're active and when they're not. And we plan to do some additional sampling this fall and also next spring to get some more data points to work with. And I look forward to answering your questions.